Hello everyone, welcome to the ITLS Academy. Today we are going to discuss the topic that is your detection of the enterococci, fecal streptococci. So why we detect this enterococci? We all know that it is a microorganism, and if it is present into a water sample, then the quality of the water decreases. Okay, and it also have impact on the health of the human being. If the contaminated water will drink by a human being, then there is a lots of problem which are occurred due to the this enterococci. That's why it is very important to detect this into a um, sample, and they are come into the water by a cross contamination. That's why it is very important. So let's start the today's lecture. The name of the lecture is detection of the enterococci in a water sample. So this enterococci are what? They are a gram positive cocci. We all know that they are what? They are a gram positive cocci. That means that they can give a crystal violet color when we detect it. When we perform a gram staining test, then it will give crystal violet color in a microscope that often occur in pair or a short chain. Okay, so this enterococci, they are always occur in either pair or either in a short chain. These enterococci are facultative anaerobic organism, and they are capable of forming spore, and they give a negative catalyst test and cocci to a egg shape. Now you can go and check in our video what is this catalyst test and how we perform it. But this enterococci give a negative catalyst test and cocci to egg shape. So this is about the enterococci. Now what the principle at which it is based: slants and Bartley medium contain a sodium azide, and this sodium azide suppress the growth of the gram negative bacteria, because we all know that when we have a sample something, now there are lots of microorganism are there. Okay, so microorganism, especially bacteria, and these bacteria are what? They are either gram positive or either gram negative. Okay, so when we perform a test and we use our Slanders and Bartley medium, then what they do, they contain a sodium azide, and this sodium azide suppresses the growth of the gram negative bacteria, suppresses the growth of that gram negative bacteria. And it also contain 2,3,5 triphenyl tetrazolium chloride, and this every ingredient have their role. Sodium azide suppress the growth of the gram negative bacteria, and 2,3,5 triphenyl tetrazolium chloride, they reduces to red formazine by our enterococci. Conformation is done by the transfer of the membrane with all the colonies on a bile acetylene azide agar. Okay, so when we have to perform a confirmation test, then we use our bile acetylene azide agar, where acetylene is hydrolyzed with 2R, so that it will form a 6,7 dihydroxy fumarine, which combined with iron third, and then it give a tan color to a black compound, which is diffuses into the medium. This is the principle at which this are worked. Now, culture media, we use our two culture media. One is your slants and barley medium, and another one is your bile asculin azide agar. So the first one, that is your slants and medium, they are what? They are used for the detection of, for the detection of enumeration of enterococci by membrane filtration technique. By membrane filtration technique. And the combination that of this we are already discussed. But the next agar that we use is a bile acetylene azide agar. They are used for a conformation. And the composition of this bile acetylene azide agar are, they contain, now I'm going to give a composition in for a 1 liter. Okay, so they contain agar 15 gram per liter. Next is your acetylene 1 gram per liter. Ferric citrate 0.5 gram per liter. Meat extract, it is around 3 gram per liter. And also meat peptone, that is 5 gram per liter. And ox bile, ox bile, it is 440 gram per liter. So this is a composition for a bile acetylene azide agar. Then we maintain the pH of this bile exude agar is around 6.6. Okay, 6.6. And this pH will be maintained with the help of the pH meter. Then after it, we autoclave it at 121 degrees Celsius for 20 minutes and 15 psi pressure. Okay, for the elimination or killing of all type of the microorganism. After that, we go to the next, that is your procedure. In a procedure, what we have to do? We clean a laminar. Okay, because we perform the experiment inside the laminar. Laminar will be clear with the help of the ethanol 70%. Then we switch on the UV light, okay, for 15 minutes, so that all the microorganism, if any microorganism are there, then it will be killed. And how we will do it, you can go and check the video related to this. Okay, the video is already uploaded in our account. Disinfect the surface of the bottle, pouch, cup containing the sample with the 70% of the ethanol, thoroughly mix with the sample by vigorous shaking to achieve a uniform distribution. Now, filter the paper requisite the volume with the help of the sterile membrane filter. And the pore size of the filter paper is around 0.45 micrometer. And then we place that filter on a slants and barley medium plate. Okay, then after that we incubate that plate at 36 degrees Celsius for 40 to 40 for 40 plus minus 4 hours in the incubator. After the incubation, we observe the plate. If it is showing a red, maroon or pink colony, that it may be a enterococci. Okay, so the first medium that we use is slants and Bartley medium, and incubation type is around 44 to 44 hour plus minus 4 hour at 36 plus minus 2 degree Celsius. Then we get a colonies in red, maroon or pink in color. Now after that, if there are presumptive colonies, then confirmation can be done by using the next agar, that is your bile acetylene azide agar, in which we transfer the membrane with a sterile forceps without inverting it into a plate which contain a bile acetylene azide agar, and then again we preheat at 44 degrees Celsius. Now again we incubate it for a 2 hours at 44 degrees Celsius, and observe the plate immediately and consider all the colonies showing tan to black color is the enterococci. Expression of the result: presence and absence of the enterococci is given after the conformation on a bile acetylene azide agar, and it is represented at present or absent per ml of a sample. Suppose that we get a 3 colonies, then 3 colonies in 100 ml. This is a result that we show. Now the next is your precaution. When we using this, when we performing this experiment, then we have to take some precaution. Sodium azide, they are what? They are highly toxic in nature and mutagenic. Mutagenic means that they are cancer causing. That's why when we are handling this media, then we have to handle it very carefully. Precautions shall be taken to avoid come in contact with it, especially through the inhalation of the fine dust during the preparation of the media. Azide also contain a media which should not be mixed with a strong inorganic acid, that is your toxic hydrogen azide may be produced. Solution which contain azide can also form a explosive compound when it come in contact with the metal pipework, for example, from a sink. So this is all about the detection of the enterococci. I hope you will understand, but if you have any query related to this topic, you can ask me in the comment box, or for a further detail, you can follow the ITLS Academy on different platforms, that is your Instagram, Facebook, Twitter, YouTube, WhatsApp, and LinkedIn. And subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. Or you can also ask your query to the helpline number that are already mentioned in your slide. That is 7080833450, and visit our website www.itlsacademy.com. Thank you for watching. Please like, share, and subscribe our channel.